Hey, welcome back. Today we're going to go through the nervous system. Of course, we should know all of this. Something that you might not know is a pseudo-unipolar neuron, which is just a dendrite fused with the axon, and it seems it's called pseudo-unipolar because it really isn't unipolar. It's actually one of the bipolar neurons. In addition, interneurons are the neurons that are only going to be in the brain, and those synapse with the efferent, which goes out into the spinal cord, and this, that, and the other. Another thing that you might not know is the enteric nervous system is a little bit separate from the sympathetic and parasympathetic system because this branch will innervate the intestines, and the intestines have both sympathetic and parasympathetic ganglions in them, so it's not just rest and digest. The same goes with the visceral nervous system, so this doesn't this includes more than just the intestines. It does include all the visceral organs in the body, so the liver, the kidneys, the stomach, and that also is in its own category, pardon me, because it has branches from both autonomic and somatic nervous systems innervating the organs. So once we go deeper, it's in medicine, in studying the body, it's not as black and white as we used to believe back in high school or even undergraduate courses. Everything is a little bit more nuanced. So there's two different types of synapses. First, we'll cover the chemicals. Synapses can be fast and slow, and that just it depends on what the chemical or the neurotransmitter that's going to be released. The cell body makes and packages the large neurotransmitters, and usually that's substance P, along with some enzymes, and then the synapse terminal will make the small neurotransmitters, which are acetylcholine, glutamate, and GABA. The terminal release of the neurotransmitters will use the transporter molecules for reuptake. After some enzyme has cleaved them from the receptors. So here is this is going to be a voltage gated, and here's the reuptake enzymatic degradation. The enzymatic degradation has to occur with some other kind of enzyme. The electrical synapse is going to be the fastest, of course, because this will conduct the action potential, and unlike the other chemical synapse, this is only excitatory because as you know, there's the action potential is an all or none phenomenon, and it only produces an action. There's no inhibition. So these are important in the body because they coordinate the neurons to fire in sync, whether it be in the skeletal muscle or in the cardiac muscles. So you can see here the same kind of deal. They're going to exocytose the neurotransmitter. Neurotransmitters are going to go across and bind to the receptors in the postsynaptic cleft, and then instead of opening ligand-gated channels, it's going to go through the gap junction channels, and then the action potential will go. In certain other cells, there's something called connexins, and those kind of function kind of like those origami flowers that you used to play with as a kid, and you were able to open it just by twisting. They do the same thing, and they also are hydrophilic, allowing water to come in. So what's important to know is that potassium is the major cation of the intracellular fluid, while sodium is the major cation of the extracellular fluid. I like to remember it as a salty banana. Inside is the banana, potassium. Outside is the salt. The large proteins are the main anions in the intracellular fluid, while sodium is chloride is going to be the main anion for the extracellular fluid. So these values are somewhat important, but just know that the internal potential is going to be negative, and in depolarization, sodium comes in, depolarizes, making the voltage positive, and then during repolarization, potassium will come back out and through the leak channels, and then it hyperpolarizes, so it goes past the threshold level, past the stimulus levels, being more negative before eventually kind of like reaching equilibrium into the resting space. So the Nernst equation will calculate the equilibrium in the electrical force that prevents diffusion down the concentration gradient. Just to boil it down, it's an equilibrium potential. You can you can plot it, as you can see here. It's linear, and this is the electrical equilibrium of the cell. Is going to be E naught minus this value. It's going to be the top is going to be it's a I don't remember what coefficient this is, but it's going to be a constant. There we go. And n is going to be whatever anion or cation that you're using value. Q is going to be your charge. As you can see here, Q is the products over reactants. Would you have to know this equation? Highly doubt it. I don't think anybody has enough time to do a Nernst equation in their head, because on the NBMEs and the boards and the USMLEs, you're not given scratch paper and a calculator. So just kind of know, in general, the Nernst equation is just for equilibrium potential. So graded potential is, just to sum it up, it's summing up stimuli, like, they build up and then it can overcome the threshold, propagating the action potential. So the graded potential are really formed in response to opening the mechanical or the ligand gated channels, and the amplitude size will depend on the stimulus strength, and it all works by summation. As you can see here, small stimuli and then large stimuli causes more depolarization, and then just more and more and more, and they add up. It has to pay the price though, because after the graded summation, the axon is going to hyperpolarize. So action potential is all or none. Suprathreshold stimulus will have the same amplitude, duration, and form as the threshold stimulus. The action potential will come from the trigger zone and then propagate down to the terminals. Spatial summation is when their neurons 1 and 2 act simultaneously on the third neuron by overcoming the threshold. So that's going to reach the suprathreshold stimulus. Temporal summation is the rapid succession of the stimuli of the same axon to cause the overlapping action potentials to summate. So like the chart before, it's just going to add on these little EPSPs in order to overcome the threshold, and then once overcome, it's going to pass the axon hillock, and the postsynaptic neuron is going to fire. But important, temporal is just one, spatial is like several different neurons. So the refractory period is the whole hyperpolarization period. There's two types, relative, which you can overcome, and the absolute. So if you decrease the amount of potassium in the extracellular fluid, that's going to bring the membrane potential away from the threshold. It's going to make it harder to repolarize the cell, so it's going to increase the time of hyperpolarization. So if this happens, like look, if the voltage gated channel is going to be inactivated, it's going to take a long time before it's going to reach the resting equilibrium. So hyperkalemia, there's going to be easy excitatory activity. The cause is going to be some kind of renal failure, and the decreased distal flow, it decreases the amount of potassium to be secreted, so it's going to just build up in the blood. Since it builds up in the blood, you can't have more, cations, so sodium instead is going to be pumped out or excreted because you don't want too much cations in the blood because it's going to acidify the blood. Primary hypoaldosteronism, pardon me, is an adrenal insufficiency. So this is going to be a problem with the adrenal glands, and in particular, there's going to be the adrenal enzyme deficiency. Second, hypoaldosteronism is going to be hyporenemia, so there's not going to be a lot of renin made, and different kinds of blood pressure medications, such as the ACE inhibitors, along with blood thinners, such as the heparin drug, will cause the problems with aldosterone secretion. And in some cases, there could be some genetic resistance to aldosterone, causing pseudo hypoaldosteronism, along with some kind of kidney disease, such as this tubulointerstitial disease. And the diuretics trimethoprim and pentamidin will also, cause a resistance to aldosterone. So with the problems with hyperkalemia and aldosterone, there's going to be some enhanced chloride ion reabsorption. Here you go. This is a nice little mnemonic on hyperkalemia. The body cared too much, so too much potassium. From intracellular to extracellular, adrenal insufficiency, renal failure. This is also another cause, excessive potassium intake. Unless you're taking potassium supplements, it's actually kind of hard to have too much potassium, because I know bananas have a lot of potassium, but you have to eat pounds of bananas a day to have too much potassium. Potassium-sparing drugs such as spironolactone will also cause the body to decrease the secretion. Hyperkalemia is shown in the ECGs. Here's the normal P waves, but in hyperkalemia, the P waves will get smaller, so these little bumps right here will get smaller, and the PR complex is going to get longer with time. So here it's getting longer, here it's very, very long, and you don't even see a P wave at all. Hypokalemia is when the electrochemical gradient is less than 90%, or 90%, 90 millivolts, and it's going to be harder to excite. This will be caused by, of course, deficiency of potassium, some kind of metabolic alkalosis, could be caused by some hormonal imbalances, an anabolic state of the body, hypothermia, barium toxicity. This usually occurs whenever it's usually in children, really, when children eat caustic substances such as batteries. And it presents with excessive diarrhea and sweating. And the problem with it is the renal failure. So there's going to be the increased distal flow. It's also associated with the Cushing's disease, Barter's, and Little syndrome. So in hypokalemia, there's going to be a slightly peaked P wave, but and a slightly prolonged PR interval. On the other hand, it's going to have an ST depression and a shallow T wave, and a very prominent U wave compared to a small U wave here and a larger T wave here. So if you're kind of suspecting hypokalemia, don't look at the PR complex. That would lead you to hyperkalemia, where you look more at the T and U waves. So neurotransmitters are neuropeptides, and they can presynaptically or postsynaptically influence whether an action potential will fire, and they're all transient. There's no permanent neurotransmitter action. So these are the important ones: acetylcholine, that's movement, cognition, automatic control, and this one's kind of like the workhorse of the body. It's excitatory or inhibitory. The monoamines, important one, norepinephrine, serotonin, dopamine, histamine, and the amino acids also work as a neurotransmitter. So GABA is usually inhibitory, glycine, glutamate and aspartate, and nitric oxide. Nitric oxide really isn't like the amino acid, but it's really important in vasodilation and some kind of like signaling. But remember, the vasodilation part. These are a little bit less important. This you will cover more in neuro. So the SNARE proteins, the neurotransmitters are going to be in the terminal, in vesicles attached to the cytoskeleton, and the SNARE proteins will mediate the vesicle fusion that contains all these neurotransmitters to bind to the presynaptic membrane. And then a protein called synaptagmin binds calcium from the calcium channel, allowing it come out. And then synapsin is phosphorylated, the vesicle will detach, and it will migrate to dock at the cells. Let's see, migrating, here it is, docking. And once they docked, the membranes will fuse and allow the cell to exocytose the neurotransmitters. So the fast chemical synapses are going to be ligand-gated ion channels, and those are direct action. The agonists are going to be nicotines. Antagonist is a drug called curare, and it just works by blocking the nicotinic receptors. The slow chemical synapses are metabotropic receptors, and they have a couple extra steps. That's why they're slower. They express the G protein coupled receptors, and then the neurotransmitter will open and close the remote channels, and that is separate from the receptors, and it can also turn on the second messenger system. So something comes in, binds here to the receptor, the G protein is going to be activated, and then it goes to the effector protein, and then it goes through CAMP or AMP, and then finally binds to the actual ion channel and allow the ions to come either in or out. And the agonist is going to be muscarine, so these are muscarinic receptors, and the antagonist is the drug called atropine. So here we go. These are the G protein linked second messengers. This phospholipase C, that is going to be involved with the IP3, and that increases the calcium extracellular concentration, allowing the smooth muscle contraction. For the muscarinic receptors, G proteins, this is G stimulatory or G inhibitory. It's going to bind to adenylyl cyclase, which turns ATP to CMP or prevents that. And then if you get CMP, you go to protein kinase, and it increases the amount of calcium available in the heart in order for the heart to contract more. So this is going to be stimulatory, or it's going to be stimulating the myosin light chain kinase, smooth muscle, of course. So this is going to be more for digestion and peristalsis. So we should know this mnemonic: kiss and kick until you're sick of the sex. So this is the sympathetic kiss. It's going to be parasympathetic. Dopamine, histamine, vasopressin. So the mnemonics, you don't, I guess, really need to know which are involved with which messenger system quite yet, but just know that there are two different kinds: phospholipase C and adenylyl cyclase. Alright, that's it for now. Thank you for your attention, and good luck studying.